presented by Caltech.
Good evening, and welcome to the Watson Lectures. Tonight, it's my distinct pleasure to introduce Professor Azita Emami for tonight's lecture. Azita received her bachelor's degree from Sharif University of Technology in Iran in 1996, and her master's and PhD degrees in electrical engineering from Stanford University in 1999 and 2004, respectively. From 2006 to 2007, she joined the T.J. Watson in 2004 at IBM T.J. Watson Research Center in 2004, and from 2006 to 2007, she was an assistant professor of electrical engineering at Columbia University. In 2007, she joined Caltech, where she is now the Andrew and Peggy Cherng Professor of Electrical Engineering and Medical Engineering. She's also a Heritage Medical Research Institute investigator at Caltech. She also currently serves as the executive officer, the department head of EE at Caltech. Her current research interests include mixed signal integrated circuits and systems, high-speed optical interconnects, silicon photonics, wearable and implantable devices for neural recording, neural stimulation, sensing, and drug delivery. She's currently the associate editor for the IEEE Journal of Solid-State Circuits, and she also serves as a Solid-State Society Distinguished Lecturer. She serves on the technical program committee of the International Solid-State Circuits Conference, and in the past has served on the technical program committee of the VLSI Symposium and Custom Integrated Circuits Conference. She was the general chair and program chair of IEEE Optical Interconnect Conference in 2015 and 2014. One of the great things about Azita, in addition to all of her achievements, is that she's a great mentor and advisor to students. I've seen firsthand many of the students, how she has helped them go from being the timid student, graduate student arriving in, and then leaving as confident contributors and peers in the field. The other thing is that she's a great citizen of the Caltech and the department, and she has served in various roles helping the department, such as her activities as the department chair, as executive officer. On the research front, one of the things that's really remarkable about Azita, and I really enjoy observing that, is that how fearless she is. She will get into new fields and new areas, and she's not afraid of tackling new areas and getting to learn them and mastering them and contributing to them. And you know, what my observation has told me is that she's the person who wants to make the world a better place, and you know, and be a very positive influence on the world around him. So tonight we are treated to a special lecture titled "The Tiny Chips for Brain, Body, and Machine Interface." Please join me in welcoming Professor Emami.
Thank you, Ali, for the kind introduction. Good evening, everyone. It's a great pleasure for me to be here and tell you a little bit about our research projects here at Caltech. So in my group, we are excited and interested about using nanoelectronics and microelectronics for the field of medicine. And we believe that we can create new devices using these technologies that can help many patients who suffer from chronic diseases such as diabetes and epilepsy. We can use these devices to monitor patients at the comfort of their homes and hopefully also send them home as early as possible from hospitals to reduce the cost and also reduce the danger of being exposed to harmful bacteria. These devices are tiny and wireless. They can be wearable or implantable, and they can help us to not only monitor patients, but also diagnose new problems. Over the past 5 years in my group, we have developed a number of such devices, and you can see some examples here. So as you can see on the top side, you see a very small intraocular pressure sensor. And then this device is going to help many patients who suffer from glaucoma. It will allow them to measure the pressure continuously anytime they need to, instead of going to the clinic and measure the pressure. Another device that you see up there is an injectable glucose sensor. This is a tiny device that is completely wireless, and I will describe it further tonight as we move through the presentation. We are also interested in neural interfaces, devices that can connect to the brain, and we can read the neural signal, and we can interpret them and decode the activity of the brain. So let me start with the glucose sensor. So as you know, there are hundreds of millions of patients who suffer from diabetes, and unfortunately, many of them need to prick their fingers every day to measure the glucose level. And as you can imagine, this is very painful, and it's not continuously, it doesn't give you continuous data. So it is very crucial to be able to replace this with continuous measurement for many patients and make it less painful. A device that we have developed is this tiny device. It's about 1 millimeter by millimeter on the surface area. It's very thin, about 100 micron. And this device can be injected right under the skin, and we believe that we can measure the glucose level in the interstitial fluid and then estimate the blood glucose from the glucose level in the interstitial fluid. So the reason that we could make this device so small, one of the reasons is that it is batteryless. And we can wirelessly power it up and also wirelessly communicate with it. We also have used nanoelectronics and nanotechnology to make this device small and efficient. So here I show more details about this device. There is an external reader at the size of maybe a clicker that I'm missing today. And so this external reader is sending the power via RF signal to an on-chip antenna. And there are thousands of transistors on this chip that helps us to measure a tiny current that is generated because of the presence of the glucose. And we basically have analog to digital conversion, and it's like a little radio that recovers the data and also sends it, communicates to this external reader. So let me first describe how the glucose measurement is done. In fact, how we measure the glucose level has not changed much over the past 50 years. It's based on oxidation and reduction, very similar to a battery. We have 3 electrodes: working electrode, reference electrode, and a counter electrode. And because glucose is cannot oxidize by itself, we use a catalyzer that is called glucose oxidase enzyme. And this enzyme will help with this oxidation, and a tiny current is generated at the counter electrode. And if we measure that tiny current, we can actually estimate the glucose level in the blood. So this picture shows the complete chip. And the interesting thing here is that we have included everything on a single device. So the electrodes are right in the middle. This is the external antenna, as you can see, and the 3 electrodes are shown at the middle of the chip. So this is about 1 millimeter by 1 millimeter in size. So you would wonder, if I make this electrode so small, then the current levels that I have to measure will be also very, very small. So the trick that we are playing here is that we create nanostructures on top of the chip, on top of the electrodes using platinum. And by doing that effectively, we increase the surface area significantly. So if you look at the surface of the chip, We remove the metal that already exists, and we replace it with platinum in collaboration with my colleague, Professor Axel Scherer. And by creating a rough surface, we can increase the current that is generated by orders of magnitude. So we start, if we have a flat surface, we get maybe less than a nanoamp. And by doing this, we get more than 100 nanoamp of current. Another significant advantage of doing this is that we increase the lifetime of the device because we can hide the enzymes in these nano trenches from the body, and then the immune system doesn't attack the enzymes as much, and the device lasts much longer inside the body. Also, by making it so tiny, we make sure that there is no scar tissue and no inflammation. And in fact, we have shown that after injection, very quickly everything heals. And for instance, in this animal test, the animals are completely okay after we do injection, and we see very fast healing of the wound or the incision that is made. So we are very excited about this device. Right now, we are testing it in mice. And we believe that it will have a huge impact, especially if we succeed to increase the lifetime of the device inside the body. One of the things that we do right now, we are creating a smart cage for animals so that we can wirelessly monitor animals or mice that are used for drug development, for pharmaceutical research. So this way we can wirelessly monitor these animals, and we don't put them under a lot of stress to take them out and draw blood, and then we get more accurate reading because the stress has shown to affect significantly the biomarkers that we are interested in. We are also developing a next version of this, which is a wearable patch for people who don't have diabetes, but maybe they are interested to roughly estimate the blood glucose level from the sweat. So that's the next version, which will not be as accurate, but can show you the trend as the glucose level in the blood changes. Another exciting domain for us is, instead of wirelessly using RF signal to power up the device, we use biofuel. We use the glucose and lactate that already exist in the body as similar to a biofuel and power up the chip in the future. So let me now change gear a little bit and also tell you about our research related to neural interfaces. These are devices that connect to our nervous system and can help patients who have neurological disorders. And these devices are already out there and are helping millions of people across the globe. For instance, a successful example is deep brain stimulation for Parkinson's disease. And this device is now very common and has helped many, many patients. Another example is a device that, again, uses deep brain stimulation to stop the seizure for patients who suffer from epilepsy. And finally, there are neural interfaces that will help patients who have a spinal cord injury, and I will talk about this further during my talk. So in my group, we were very interested in this domain, and we started with seizure and epilepsy. And one of the main reasons for doing that is that, first of all, it's a very common disease. And another major problem with it is that about 1/3 of people who suffer from epilepsy cannot benefit fully from the existing drugs. For these patients, there are two options. Some of them go through a surgery. They go to the hospital for many days. They monitor them. They open the skull, and they put electrodes and monitor them. And then they may remove a small section of the brain where the seizure starts. Or they can actually, what we are envisioning is that we can predict the seizure and then stimulate again, perform deep brain stimulation to stop the seizure. So let me talk a little bit more about this kind of devices. There are existing devices that try to do this. They try to stop the seizure by deep brain stimulation. One of these devices, which is FDA approved, is called VNS. It's an open loop device that the patient decides when they want a stimulation, and they click a clicker, and the stimulation happens. Another device tries to do this in a closed loop manner by predicting the seizure and performing a stimulation. But unfortunately, this device is very large and also not very accurate. In my group, we wanted to tackle this problem, and we wanted to predict the seizure as early as possible, and then create a complete closed-loop system that is very small, it's miniaturized, and it's very efficient and completely wireless, which doesn't hopefully need a battery in the future and can last very long. So before I start talking about the details of this device, I would like to tell you a little bit more about neural interfaces. A very important component, as you can imagine, is how we interface an electronic device with biology. In the case of electrodes, we have a number of options, and each of them have certain pros and cons. For instance, EEG, I'm sure many of you have heard about it, is completely non-invasive, and it sits outside of the brain. But at the same time, it doesn't give you much information. It gives information about collective activity of the neurons. Another option which gives more accuracy is called ECOG, or intracranial EEG, where you put the electrodes, which are flat electrodes, right under the skull. So this is a great compromise that is a little bit more invasive, but at the same time provides more accuracy and provides more information for studying the brain. And finally, there are these penetrating electrodes that go into the cortex. As you can imagine, these are very invasive, but at the same time, they provide amazing level of information at the neural level and cell level. And they have been great tools for studying the brain. So in terms of seizure, because we don't need that level of cell accuracy, we can use IEEG or ECOG. And studies have shown that if we make these tiny electrodes, as you can see, these are electrodes that sit on top of the brain. So as you can see, there are larger electrodes and there are tiny, smaller ones. And research has shown that if we use many of these tiny ones, we actually can predict the seizure much earlier, which is very beneficial. The earlier you predict the seizure, you have a better chance of stopping it or even taking medications to avoid the seizure to happen. But at the same time, that means that if you have these tiny electrodes and many, many of them, and you're sampling at a very high rate, it means that you're generating a huge amount of data. So this brings about a big challenge that what do we do with all this data, especially if we want to have a closed loop system. And if we want to make it very low power and wireless so that we avoid batteries. So here is where electrical engineering and signal processing and electronics, they all can come together to solve such a problem. In fact, we used a technique called compressive sensing, which is known to reduce the number of samples that you need compared to standard approaches given a bandwidth of a signal, because neural data is actually sparse. So we use compressive sensing to be able to reduce the hardware complexity and reduce the power consumption and size of this device. So we implemented this chip, again, in a standard CMOS technology, which is very, very small, again, about 1 millimeter by 1 millimeter in size. And we tested this in collaboration with UCLA Neurosurgery Department, where we could actually predict the seizure. And then this device also has a stimulation channel, and we could stimulate, as you can see, this is the stimulation current to stop the seizure. So this gave us a significant reduction in area power product, which could lead to a device that can be implantable and wireless. So this was a great experiment for us, but at the same time, we realized that in the lab, every time that we want to do the test, we have to adjust the threshold levels and many parameters, and also for different mice, the parameters would be different. So we organically and kind of naturally went towards more adaptive systems, and we started looking into machine learning algorithms that helps us to build a system that can be trained for any patient, and then we set the parameters, or we can update the parameters as we train it again, and basically have a more efficient system. But as you may know, you have heard about machine learning or artificial neural networks. A lot of these approaches are extremely hardware inefficient, meaning that they require a lot of components that is very hard to implement on a single chip that is low power. So for a start, we actually looked at a decision tree as an element of classification, or making a decision whether a seizure is happening or is not happening. So a decision tree has many nodes, and each node will say a yes or a no to you, and you can assign a certain channel or a certain feature to each node. Features in terms of neural data means, for instance, power in different frequency bands, or correlations, or time variances. So there are many, many features that we can assign to neural data. And during the training, we can decide what feature and what threshold level we should assign to each node of this tree. But these trees are considered weak classifiers. They're not very strong classifiers. But one thing that it has been shown that mathematically that we can do is that we can incrementally increase the number of trees. This technique is called gradient boosting. We add the trees incrementally to get gradually better results. And then we combine the result of all the trees together and make a decision whether, for instance, the seizure is happening or the seizure is not happening. So we use this technique, and we actually showed that for seizure detection, we don't need too many trees, or we don't need trees that are very long. So basically, we can use shallow trees, and we can use 8 to 10 trees to get very good results in terms of seizure prediction. So this plot shows the number of trees that we need and the depths of the trees that are required to get good results in terms of sensitivity and specificity. So after we developed the algorithm and we showed how many trees we need and how we have to program these trees, we implemented these in hardware. And we came up also with interesting, my postdocs and the students came up with interesting techniques how to implement this kind of algorithms on chip that is power efficient and also area efficient. So this was implemented again in a CMOS technology, in a 65 nanometer CMOS technology with very, very small area using 8 fully programmable decision trees, and that can support many, many number of features that are required in these techniques. It also consumes a low level of power. So this is, again, a very important feature for future implementation of these devices. This device overall consumes less than 100 microwatts when all the trees are operational and we are at the maximum power consumption. So we used, as I mentioned before, this was a version of a machine learning-based neural interface. And from this project, we realized that we can extend it to other types of brain-machine interfaces. So at Caltech, some of you may know, Professor Richard Anderson has been working in this domain for many years. And the main idea here is that for a patient who suffers from spinal cord injury and paralyzed, to implant electrodes in the brain. These are, in this case, because we are trying to do fine movements of a robotic limbs by reading the brain activity. In these cases, we have penetrating electrodes, which gives us a lot of information. At the same time, the reward is very high. It's a very invasive technique, but at the same time, provides a lot of flexibility and freedom to the patient. As you can see, this patient then, by thinking, can move the robotic limb. So I'm going to show this video. So here, as you can see, the patient is, just by thinking, has trained his brain to move the cursor as the cursor moves across the screen. And this is an amazing achievement. So again, in Professor Anderson's group, there has been many years of research to look at different areas of the brain to see where to put these electrodes. And the patient is trained to, by intention and attempting to move a cursor from the middle point to a certain point. And we use this data to basically train our algorithms and predict the movement. So again, this is a very complex system. We are dealing with hundreds of electrodes that are sampling at very high data rate, and a huge amount of data is generated. So there are a lot of signal processing going on to be able to estimate the movement, the speed, and the location of the cursor or the robotic arm. But at the same time, while this is remarkable, the existing techniques are usually relying on a very famous approach or control theory filter that is called Kalman filter. So this filter, while provides good control, at the same time, it has a lot of challenges. When you have these penetrating electrodes, these electrodes move around day to day, and sometimes we lose electrodes, sometimes the electrode comes back in, their encapsulation, and so on. So there are a lot of non-stationary conditions. There are also a latency in the loop and inaccuracies. Maybe if you look at that video, you see that it's not exactly accurate. And there are a lot of high misclassification rates. And the system overall deals with a lot of noise and modeling difficulties. Also, right now, the patient is connected through these wires to a computer, and all the control is done using a processor, which is very high power, and it's very difficult to implant such a device. So our goal has been to, again, look at machine learning algorithms for decoding the kinematics or the movement from the brain data. So if you look at the artificial neural networks, this is a very simplified network that many of you may have seen. Basically, the artificial intelligence that you hear about is mainly based on an architecture like this. This is a feed-forward neural network where the data moves forward. But for data such as brain data, we basically highly require the memory in the data. So the data is like a sequence. Therefore, it is very important to add some internal memory to the neural data. This kind of networks are called recurrent neural networks. And they're very powerful when you're dealing with the data that has history or sequence embedded in it. So in my group, we looked at the hardware complexity of such algorithms. And if you just go for the standard algorithms, you quickly realize that you need many number of nodes and many number of layers, and this is what is called deep learning. So we decided to look at this specific application, and in fact, we were able to come up with a new algorithm that we call it dynamic recurrent neural network, where we add an extra state to the system because of the neural data, the properties of neural data, and also we add history to the neural network. So by doing this, we showed that we can make our neural network much simpler and easier to implement in the hardware. At the same time, the performance of this dynamic neural network was superior. For instance, this shows the estimate of y when we are estimating xy, when the patient is thinking about moving the robotic limb. And the red is our estimate from our algorithm, and the black is the actual location. So as you can see, we are getting very good results compared to, for instance, this is the standard Kalman filter, or this is the linear models that sometimes are used. Because this is a nonlinear model, and it's an artificial neural network, we are getting better results. So again, another way to evaluate the performance of this approach is to look at the correlation coefficient between the actual location and the estimated location. And this one is the dynamic recurrent neural network that we have developed, and these are the standard approaches. So again, what is really important is that in our technique, we use only 5 neurons or 5 nodes and history of 10, which is extremely important when we are trying to implement things in hardware. So right now, that's our goal. Our next step, this is a very new project for us. Our next step is to actually implement it in hardware, and one day be able to remove all the wires that are coming out of the brain of the patient and remove the computer that the patient right now needs to be connected to. And as you can imagine, that doesn't provide any mobility to the patient. And hopefully by creating this new device, we can add mobility to the condition that we have right now. So now let me tell you a little bit about another project in my group. As you know, if we have these tiny devices that one day we can send them to the body to fix problems or identify problems, we also need to know where they are. This is, you may, if you're old enough, you may remember this movie, Fantastic Voyage, based on the book by Asimov, where a tiny submarine is shrunk to a very small size and is sent inside the body of a scientist to destroy a blood clot. At the same time, the submarine communicates with the external world, and they know where they are. So if you are younger, like my kids who are here, you may remember a magic school bus when the bus is shrunk and is sent inside the body to investigate. So we might be able to do that one day, to send small devices inside our body and send them to fix problems. But it means that we should know where they are exactly and be able to communicate with them. And that turns out to be actually a very difficult problem to solve. If you use techniques similar to GPS using RF triangulations, you don't get much accuracy because the time of arrival of the RF signal in the body depends highly on the body composition. So whether the wave is traveling inside the muscle or inside the fat or bones, the time of arrival will change and the estimation will be difficult or inaccurate. And we already have devices that we would like to track, for instance, smart pills that are common. It would be great to know where they are in the body as they are moving inside, if we one day want to also add sensing to it. So in my group, we decided to look at what is the high-resolution imaging techniques that are out there. One of the highest resolution that you can get is to use MRI. And in the MRI system, you can get resolutions that are as good as 100 micron or below. So the way that MRI systems work is that you have a very strong magnetic field that align the nuclear spins. The atoms inside the body behave like a nuclear spin, and you can align them with a very strong magnetic field. But then if you also create a gradient, a known gradient in this magnetic field, and you apply an RF signal, after you remove the RF signal, different atoms at different location or different nuclear spins will resonate at a different frequency. This is called the precession rate or Larmor frequency. So we thought that maybe we can build our chips, these sensing devices, in a way that behaves similarly to real atoms inside the body. And we call them addressable transmitters operated as magnetic spins or atoms. But these are artificial atoms. These are tiny chips that also behave the same way. So first, if we apply an RF signal, we lock all of them to the same frequency. And then when we remove that RF signal, each of them will resonate at a different frequency. And this is done by, again, creating a magnetic field that has gradient. So different chips at different locations will measure a different magnetic field. And then they shift their frequency of oscillation accordingly based on the measurement that they have. So we were able to build this chip again in CMOS technology. It has an on-chip magnetic sensor. It has an on-chip oscillator that can lock to an it can wirelessly lock to an RF signal. As you can see, this is an antenna that receives the RF signal, and it locks to the incoming frequency. And then the internal oscillator, after it's shifted based on the measurement that we have, sends an RF signal out, and we can measure it, and we can decode the location by looking at the frequency of oscillation. So imagine if this chip is moving inside the body of this mouse. As it moves, it oscillates at different frequencies, and we can measure that. So we basically tested this device. We first tested it on the bench, and we moved the device and wrote California Institute of Technology, as you can see, with very high resolution, close actually to the MRI resolution of about 100 micron. And we also did animal tests. We were able to send this in the body of a mouse and move it and see how we can locate this. So after we published this, it was very interesting because there were many applications that we had not thought about this, but after we published it, the physicians and surgeons at different hospitals saw our work and contacted us with possible application. Imagine anything that moves in the body, or if we have devices that move around, then we can locate them very precisely. So one application, of course, that is clear is smart pills. We first thought that we need to add some sensing mechanism to this device. So as they, for instance, move inside the GI tract, we measure something such as pH or other biomarkers, and then also measure the location. But now we are collaborating with a doctor in Harvard Medical School, because he told us actually that even if you don't measure anything, if you tell us how fast things move in the GI tract, is very, very valuable. Because there are many patients actually that suffer from chronic constipation or other diseases, and it's very important for the doctors to know where the slowdown is, or to monitor the speed that things move inside the body, inside the GI tract. Currently, the way that this is done is that the patient drinks a number of these markers that are just plastic markers. They are passive. And then the patient goes back maybe 8 hours later or the following day, and there is an X-ray that shows where the devices are. If you look very carefully, you see these devices inside the body of the patient. But of course, this just gives you one snapshot, and also it requires X-ray, which is harmful to the patient. So instead, we are foreseeing that we build these atoms devices that the patient can drink a number of them, and all we need is an external vest that creates the magnetic field. Using electromagnets, we can control the magnetic field and create the gradient that we need, and in fact, we have done that already, and the next version of the chip is being fabricated to be able to use for this kind of application. Another application that, again, they came to us was related to high-precision surgery. This is one example. There can be many surgeries that this device can have impact in that domain, but this is a surgery related to orthopedic, and where if you want to put a nail, a titanium nail inside the leg, after they insert the titanium nail, they need to fix it inside the body with a screw. And the implant, the titanium implant, has a small hole, and the doctor needs to align the screw with that hole and fix it in place. So, and that requires quite high resolution of about 100 or 50 micron. Right now, the way that it's done there, the patient goes through many X-rays during the surgery, and it takes many hours to perform the surgery, and highly depends on the expertise of the surgeon. So what we want to do, and we are actually very close to do, is to add these atoms devices to the implantable titanium nail, and also add another one to the surgical tool or the drill bit that brings the screw close to the hole. So this way, and we can show this on a monitor in a 3D, this way the doctor, the surgeon, knows exactly where things are in respect to each other, and they can perform the alignment much faster. So this can also help for other types of surgery, any organ that moves around a lot during the surgery, such as liver and lungs. These are other areas that, again, we have talked to surgeons, for instance, in City of Hope and other places, that think that this device can help them significantly in the future. So this is where we are right now. We are building an external vest, and we also building the chip, but what matters is that in the future we can scale it up. This is the advantage of CMOS technology, that if we build many, many of these devices, they will become very low cost, and many people can use them for a variety of applications. And hopefully we can translate these to actual devices that physicians and patients can use in the future. So I would like to finish my talk by acknowledging my group. There are many students and postdocs who worked on these projects that I talked about today. These are some of the members of my group. These are some of the previous members of the group and the current members of the group. The amazing thing is that a lot of undergraduate students contributed to these projects. They are very interested in medical devices and have been very active members of my group. So at this point, I would like to stop and answer any questions that you may have.